Good morning, good afternoon, and good evening. This is the video of the Biology Spotlight, where we shed some light on a lesser known scientist in history. My name is Jason Shoemaker, and today I'll be talking. Hey! Rude! Uh, well, I guess my face isn't really necessary, I suppose. Fine! Today, we're talking about enzymes. More specifically, we'll be talking about the scientist who discovered enzymes, a chemist by the name of Anselme Payen. He was born on January 6th, all the way back in 1795. Yeah, that far back. In Paris, which is in France. Payen was the son of a business owner, one who owned several chemical production factories. Anselme began studying chemistry with his father at a young age and continued to study chemistry with friends and colleagues as a young adult. When Payen was only 20 years old, he was promoted to head of his father's borax production plant. Almost right away, he made great discoveries in his occupation. He was able to invent a refinement method that was much cheaper than all the other competitors, putting his plant at the top of the production food chain. Not but 5 years later, Payen created an effective gas mask filter due to his research in sugar and sugar beets, a filter concept, I might add, that is still used in filters today. It was not until he was 38 that he discovered the first enzyme diastase. The pattern of providing the suffix ase to enzymes was started by Payen. 2 years later, he became a professor concentrating exclusively on research at the École Centrale des Arts et Manufactures, or Central School of Arts and Manufactures. Give me a break. I'm not French. Payen went on to be the first to isolate cellulose, establishing the naming system for carbohydrates, as well as discovering pectin and dextrin. He was big on organized naming systems. Quite a legacy, actually. Anyhow, Payen lived a long life of 76 years, but he did not actually die of old age. In 1871, the Franco-Prussian War was in full swing. During the siege of Paris, Payen refused to leave the city when the Prussian army advanced. Payen was killed in the attack on May 12, 1871. But we aren't here to focus on his death. We're here to focus on his life, and in particular, his discovery of the enzyme diastase. Diastase is the name. Okay, what is going on with these slides? They are coming in from random directions all over the place. Don't do it. Don't you do it. Okay, real mature. Can you pick a direction, please? Maybe from the left? Thank you. As I was saying, diastase is the name of an enzyme we now call alpha amylase. This enzyme breaks up complex carbohydrates into smaller sugars that can be more easily metabolized. During Payen's research into sugar and sugar beets, he developed a chemical from malt extract that catalyzed the starch to sugar conversion. This is a fancy way of saying he was able to see the enzyme at work. The enzyme comes in, makes contact with the sugar, and then separates it into smaller pieces. Enzymes are a truly fascinating substance. Without them, we wouldn't be able to survive. They help lower the activation energy required, or the amount of force needed to break apart a substrate, basically a molecule of some kind. In other words, enzymes make it easier for materials in your body to break down, allowing you to digest food and acquire the nutrients. In fact, according to Dr. Richard Wolfenden, if enzymes didn't exist, it would take 2.3 billion years for our bodies to complete vital biological transformation. Enzymes are one of the most critical components to the human body. This microscopic substance is capable of turning an unfathomable amount of time into mere milliseconds of work. Without these incredibly amazing catalysts, life would not be possible. Back to enzymes and what they do. There are, of course, other functions of enzymes, like DNA replication and RNA transcription, but the easiest function to understand is metabolism, how our bodies can break down food and get nutrition from it. There are many types of enzymes, like lactase for lactose and amylase for carbohydrates. The reason for this, the reason for so much variety, is because an enzyme is very specific. The binding sites where the substrate can attach to an enzyme only correlates to a specific shape, like Lego pieces fitting together. Once the matching substrate finds its little slots in the enzyme then helps break it up into smaller pieces. Our knowledge and discovery of enzymes has been one of the foundational building blocks for all that we know about metabolism and the life cycle itself. Amazingly, our study and discovery of enzymes is just getting started. Scientists have discovered enzymes that can break down plastics. With careful application, there may one day be a viable solution to the global plastic crisis. Though there is so much more to discover, it is important not to forget who pioneered the first steps in this field. Anselme Payen and his starch experiments.